Hi dear participant, we are in SWAM course in our week 7 and lecture 5. Course name is Introductory Animal Physiology. Module name is Reproductive Physiology of Female and title is Gonadal Development, Anatomy of Female Reproductive Tract and Follicular Development. Hi, I am Dr. Manish Kumar, Assistant Professor in the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Sciences, Institute of Agricultural Sciences, RGSC, BHU. Today we will be starting the new topic, that is reproductive physiology, and we will be covering in this the female reproduction. So in this topic we will be covering reproductive system, sexual differentiation at different level, functional anatomy of female reproductive tract and follicular development. So let's start with the reproductive system. Animal may reproduce sexually or asexually. Those first cell on the earth and indeed all extant prokaryotes reproduce by mitosis, a cloning process in which one cell divides to give rise to two genetically identical cells. Eukaryotes cells also reproduce this way, but eukaryotes also evolve an innovation that is called as sex. Budding or fission, in which the animal produces a copy of itself without embryonic development. Example, some sea anemones, they split into two halves and produce the two genetically identical individuals. Parthenogenesis is process of fertilization in which that one unfertilized egg will be developed into two individuals. Sexual reproduction, which depends upon the union of male and female gametes, that is reproductive or germ cells, each with the single set of chromosome, that is haploid, to form a new individual with unique diploid set of chromosomes. So this is a sexual reproduction. Sex is a total morphological, physiological and psychological differentiation between the male and female animals. Reproduction is essential for continuity of species in living organism. Asexual reproduction, the progeny genetically identical to the parent. In the asexual reproduction, the progeny which forms is genetically identical, whereas sexual reproduction produces the progeny which is genetically dissimilar to the parents. In sexual reproduction, there is random segregation of the genes during the meiosis of the gametes, recombination of the genes at the fertilization, segregation and recombination of the gene provided for the genetic variability and genetic repair. Sexual differentiation, the two sexes can be recognized by their morphological features. The complete sexual differentiation includes the total morphological, psychological and physiological differentiation that distinguish a male and female individual. With the union of sperm and ovum, there is formation of the zygote, which undergoes several mitotic division. The period of embryo consists of the time from the blastocyst development until the differentiation of the organ system in the embryo. The sexual differentiation takes place in consecutive steps with chromosomal, gonadal, somatic and psychic sex differentiation. Coming to the chromosomal sex differentiation, chromosomal sex is determined by a single pair of sex chromosomes. The sex having the similar sex chromosome is homozygous sex, whereas one with the dissimilar sex chromosome is called as heterozygous sex. Among all the mammals, the female is a homozygotic animal. Chromosomes will be XX, whereas male is a heterozygous, that is XY. Whereas same in the poultry or hens, the female will be heterozygous, that is ZW, and male will be homozygous, that is ZZ. Chromosome will be there. The chromosomal sex of an individual is determined at the time of fertilization itself. Coming to the gonadal sex differentiation, the primitive gonads has all the cellular elements to give origin to testicle or an ovary. Differentiation of the sexual characteristics depend upon the quantitative relationship between the male and female determining gene and their integration with the internal environment. Genes associated with the female characteristics are believed to be located on the X chromosome. Thus, XX chromosome are necessary for differentiation of the normal ovary. Gene for the male development located on the Y chromosome. So, the presence of the Y chromosome is responsible for the development of the testes. Gonadogenesis begins with the formation of the genital ridges. Primordial germ cells migrate from the yolk sac endoderm to the genital ridge. The gonads at this stage are still sexually bipotent and consist of an inner medulla and outer cortex. Primordial germ cells invade the medulla to form the primary sex cord. At this time, gonadal differentiation takes place and the gonads loses its bipotency. When the gene of masculinity of an XY individual prevail, the medulla persists, cortical development is inhibited and testes develop. When the gene for the femaleness predominates the XX embryo, the medulla is inhibited and the cortex develop. The persisting primordial germ cells in the epithelium invade the cortex as secondary sex cord and ovary develops. During the indifferent stage of the gonad is potentially capable of either testis or ovary formation. The ovary or testis begin the primary sex organ, but once there is a differentiation of the gonad is complete, the individual or organism can be distinguished as a male or female. Somatic sex differentiation. In an indifferent stage, there are two duct systems, that is, one is Mullerian duct system, second one is Wolffian duct system. When the testis has formed, the female, that Mullerian duct system will regress to vestigial rudiments and the male Wolffian duct system will be developed under the influence of the secretion from the newly formed testis, that is, means testis of the fetus. From these two duct systems, eventually there is a development of the complete male or female reproductive organs. Thus, at the time of birth of individual has morphologically differentiable identification either male or female reproductive organ. Now coming to the psychic sex differentiation, and this is the final step of the sex differentiation. At the time of birth, despite of having either male or female reproductive organs, an individual cannot behave, feel or act like male or female individual. The organism does not have a sex drive or libido at this stage. The psychic sex establishment is the final step in the differentiation of the two sexes. The psychic sex, the sexual behavior is basically under the control of gonadotrophin and gonadal hormones secreted in proper amount and specific ratio. Now coming to the follicular development, we will be talking about the how follicles develops. Ovarian follicle, the oocytes and its encasing cells, follicle cells at any stage of the development is called as ovarian follicle. Ovarian follicles are considered as a basic unit of female reproductive biology. Follicle provide congenial environment for the growth and maturation of the female gamete and readiness for its subsequent fertilization after ovulation. Let's talk about the embryonic development of the ova and the follicles. How it develops during the embryonic stage of the life or fetus. Early in the first week of the embryonic development, primitive large germ cells called the primordial germ cells are present in the yolk sac. These primordial germ cells migrate by amoeboid movement from yolk sac to the genital ridge. This migration occurs in 35 days of gestation in the bovine. After a few days, the gonadal sex can be distinguished by formation of superficial tunica albuginea and peripheral location of the oogonia in the primitive ovary. This occurs 45 days of the gestation in the bovines. These oogonia multiply by mitosis after the sexual differentiation and enter the prophase of the first meiotic division when they are called the oocytes. The number of oocytes produced may be determined by the level of FSH in the embryo. Oogenesis is the transformation of the oogonia into the oocytes. It is called as oogenesis. Oogenesis is completed before or shortly after the birth in all domestic animals. In this stage, the ova are surrounded by single layer of follicular epithelium, and these are called the primordial follicles. Thus, at birth, all female animals are born with their full complement of oocytes in the primordial or older follicles. In these are never replenished but only decreases in the number during the life of the animal. Ovarian follicle in the mammalian ovaries contain two different pools of the follicles. One is non-growing pools and another is growing pool. The non-growing pool contain the primordial follicle, whereas growing pool contain the primary, secondary or tertiary follicle. The formation of Graafian follicle from the pool primordial follicle during the estrus cycle is called as folliculogenesis. The formation of the follicle pools develop into the primary then secondary follicle independently of the gonadotrophin. Growth of the secondary follicle to an antral follicle is increasingly gonadotrophin dependent, with the ovulation being the entirely gonadotrophin dependent. That means ovulation will take place only when the LH surge will be there. If LH surge is not there, the ovulation cannot take place. Now let us talk about the primordial follicle, how it looks like. Primordial follicles are non-growing inactive follicles and it consists of an oocytes surrounded by single layer of flattened squamous epithelial cells, whereas primary follicle are active growing follicle in which an oocytes is surrounded by single layer of cuboidal epithelial cells. As development proceeds, the proliferation of the granular cells not only increases the number of the cells but also granulosa cells layer around the oocytes. Secondary follicle, also called as pre-antral follicle because of the absence of antrum. Other name of secondary follicle is pre-antral follicle because there won't be antrum in the follicle. That is, antrum is fluid-filled cavity. The follicle having the two or more than two layer of the granulosa cells but not more than 6 layer. It may have the 2 or more than 2 layer of the granulosa cells but not more than 6 layer of the granulosa cells. All thecal cells layer also develops around the membrana granulosa. Zona pellucida is absent in the secondary follicle. Tertiary follicle are characterized by the presence of the 6 or more than 6 layer of the granulosa cells. Well differentiated thecal cells layer will be there, which will be divided into theca externa and theca interna, and zona pellucida will be there. Zona pellucida is a glycoprotein layer secreted from the granulosa cells and lies between the plasma membrane of the oocytes and the membrana granulosa. Granulosa cells maintain contact with the oocytes through the zona pellucida by means of cytoplasmic process. Both theca interna and theca externa differentiate from the interstitial stromal cells immediately surrounding the follicles. Both layers of the theca cells are separated from the membrana granulosa by a basement membrane. Granulosa cells secretes the follicular fluid which accumulates between the granulosa cells in the form of separate small fluid filled cavity. These small fluid filled cavity fuse to form a single large fluid-filled cavity that is called as antrum. This is because the tertiary follicles are also called as antral follicle or vesicular fluid because of the presence of the antrum or fluid in the cavity. Now coming to the Graafian follicle, or it is also called as pre-ovulatory follicle because Graafian follicle will ovulate. That's why it is called as pre-ovulatory follicle. Graafian follicle was named by the Renzer de Graaf, a Dutch physician and anatomist who discovered the ovarian follicle in the rabbit first time. It contains single large antrum and a cluster of granulosa cells surrounding the oocytes called as cumulus cells, and extension of the membrana granulosa. So here you can see the test structure. This is a Graafian follicle. You can see very much this is looking like transparent and it will rupture. The innermost layer of the cumulus cells is termed as corona radiata. A cluster of cumulus cells located at one side of the antrum on which oocyte rest is called as cumulus oophorus or discus plesurus. The fluid present in the antrum is called as liquor folliculi or viscous fluid. Now coming to the functional anatomy of female reproductive tract, we will be talking about what are the different parts of the female reproductive organs and what is their functions, how they are made up of, and then species variation we will be talking in this. So if you talk about the female reproductive system, consists of a pair of ovaries, duct system, a pair of oviduct or fallopian tube, there is uterus, cervix, internal vagina, external vagina and the vulva. External genitalia includes outer vagina, vestibular gland, clitoris, labia majora and minora. Hymen may present at the junction of the outer and inner vagina. Primary sex organ, that is ovary in the female, it is involved in the gametogenesis. Accessory sex organ includes the fallopian tube, uterus, cervix, vagina, vulva. These are the accessory sex organ which is essential for the reproduction but not for the gametogenesis. Gametogenesis is mainly taking place in the ovaries. So let's talk about the ovary. Ovary is a primary sex organ and it serves as a both gametogenic as well as endocrine functions. The ovaries shapes varies according to the species and stage of estrus cycle. The functional ovary of a polytocous animal, that is like sow, bitch and cat, has the several follicles or corpora lutea which gives the appearance of a cluster of grapes. In a monotocous animal, cow, ewes and mare has an ovoid shape of ovary. In a mare has a kidney shaped ovary because of the presence of the ovulation fossa, which is a site of ovulation in case of mare. The outermost surface consists of the single layer of the germinal epithelium which covers the entire ovary, except in the mare it is limited to the ovulation fossa only. Second part, that is oviduct. Uterine tube or fallopian tubes are paired convoluted tubes which has the unique functions of conveying the eggs and sperm in opposite direction. It connects the ovary to the uterus. Oviduct functionally divide into 4 different parts, that is, first one is fimbriae, infundibulum, ampulla and isthmus. Fimbriae, it is the ovarian end of the oviduct with the fringed edge. That end portion will be like fringed, finger like projections will be there, that completely covers the ovary in pigs. Ovarian bursa covers only ovulation fossa in case of the mare. Fimbriae moves freely and sweep on the surface of the ovary at the time of ovulation. Infundibulum, it is a funnel shaped second portion of the oviduct. Size varies with the age and the size of the animals. Ampulla forms more than half the length of the oviduct, connects the infundibulum to the isthmus. Isthmus connect the uterus at the uterotubal junction. Let's talk about the function of oviduct. Oviduct is involved in the picking of the ovulated eggs from the ovarian surface. Oviductal secretions provide the capacitation of the sperm, conducive environment for the fertilization and nourishment of the young embryo, protection of the oocytes and sperm and young embryo from the attack by the maternal immune system, for cleavage of the young embryo by secretion of the cleavage inducing protein. So, this is the function of the oviduct. Ciliary movement, muscular contraction and fluid flow help in the denudation of the follicular cells from the ovum, transport of the zygote into the uterus, prevention of the tubal implantation. Oviduct transport eggs and sperm in opposite direction at about the same time when the fertilization need to takes place. Coming to the function of the uterus, uterus has the body and two uterine horn, that is called as carnua, and it serves as the following function. It is a myometrial contraction occurs at mating, causes the transport of the sperm from the site of ejaculation to the site of the fertilization. After estrus, uterine contraction directed towards the cervix, which helps in the transport of the zygote from the oviduct into the uterus for implantation. Uterine contraction keeps inhibited by high progesterone levels during the gestation. At the end of the gestation, uterine contractions that helps in the parturition. Pregnancy could be housed in the uterus since myometrium has the ability to grow enormously. Functions of the uterus: endometrial secretion induces the capacitation to the at estrus, stimulate the sperm metabolism, provide the nourishment to pre, early and post-implantation embryo during the luteal phase. Example, co-embryo live freely and proliferate extensively till 30 days post-estrus. Estrogen and progesterone receptors in the secretion modulates sensitivity of the myometrium and proliferation of the endometrial epithelium and glands. Uteroferin found in the Eve's uterine secretions also promotes the embryonic growth and implantation. Maintenance of the or regression of the corpus luteum is being done by uterus. Sends the signals to the corpus luteum about the presence or absence of the conceptus. The uterus is one which produces the PGF-alpha in the absence of the conceptus, so that Cl are being regressed or lysed. Cervix. Cervix is a thick walled sphincter-like structure, separates the vagina from the uterus. Lumen is a narrow and exhibited the annular rings. Very prominent in a cow and Eves forms the interlocking rings. In the sow, rings are corkscrew shaped. Function of the cervix: it involves in the transport of the spermatozoa from the cervix to the uterus, and it also acts as a reservoirs of the sperm, and it will release the sperm slowly to the uterus. It also acts as a sperm selection center. It will separate the dead or weak spermatozoa. It will allow only the live and progressive motile spermatozoa to pass into the uterus. Protection for the upper reproductive tract: it protects the upper reproductive tract by closing the cervix so that any infection or contamination should not enter into the uterus. It helps in the parturition. At the time of parturition, cervix relaxes tremendously and allow the fetus to come out from the uterus. Cervix during the pregnancy, it is being tightly closed and it seals so that bacteria, virus, any infection or any spermatozoa cannot enter into the uterus, so that pregnancy can be maintained. Vagina. Vagina consists of two parts, that is outer vestibular portion and posterior or internal portion. Wall consists of surface epithelium, muscular coat and serosa. No mucosa is there. Vaginal smear could detect the phase of estrus cycle in the rat and beach only. The functions of vagina: it is a female copulatory organ in which semen is deposited. It acts as a semen reservoir for the cervix. It is important for the psychosexual satisfaction, sperm transport, and absorption of the seminal plasma. Along with the cervical mucus, buffer the pH of the vagina. A specific smell of the vaginal secretions in cow at estrus identified by the bulls and the dog. Thank you.